Or if that's too big, but try it.
Maybe like,
Isn't working in there. Oh,
yeah.
there we go. Ready?
Yeah, yeah, sure. Yeah. It's in the tube, and there it is. Cool. I don't think that's swimming anywhere. Great.
That worked well.
Jar number 1.
Okay.
And indexing to jar 2 to prepare for the next sample. Nice sample.
Thank you.
And I'm going to retract that drawer if that's okay with you.
Go for it.
Stowing the drawer. I'm surprised it all stayed together. It must have been slightly like more flexible than previous ones.
Seemed like it just kind of bent right into the suction hose.
Yep.
Yeah. Okay. Craft looks good. Hydraulics off.
All right, yeah, so we just grabbed a sample of that white Paragorgia specimen with our suction sampler because the 4 bio boxes of the ROV were already full.
Okay.
So we've got quite a fairly heavy haul of biology to take back home with us.
Turn my lights on, see what it looks
Sure.
like?
I'm going to say we're pretty much full up
light
here
art
for specimens,
on
but we
cereals? Here
have
we
collected
go.
a lot of really interesting and
Is that better or worse?
collections today. So
You could turn your head to
we have our
starboard a
work
bit.
cut out for us.
Yeah.
I'll back up.
And we're
so
really
not so stretched out.
hoping that these specimens will be of really great use
Yeah.
to other scientists that are
What
actually
do you think, Dan?
studying these animals. So us
lights off better?
specifically
I think so.
aren't
Yeah.
studying these
We're gonna
animals.
bring 'em
We're
back off, Art.
collecting
I
these
think we can
animals
see the
for everyone, for the
slope a little
community.
better. Yeah.
It's not just for us.
Turbid
And that's
water.
what makes our operation here aboard the Okeanos Explorer so different than most scientific research vessels.
Probably bring Sirius a little closer to D2.
We're
What do you
here
think?
exploring and
Yeah, I'm
trying
gonna
to
turn
collect some
your
baseline
heavy
data,
bumper.
trying to help make new discoveries, and work together as a community to learn more about our deep sea.
Maybe like a
Pilot,
15 meter.
Yeah.
would it be possible to pirouette around this coral and then see that one behind it?
Sure. I think we're going to need to get a ship move in. Give
Okay.
ourselves a tether to do that.
15
But
meters
that
at
should be
1.5.
possible. Sure.
That sound good, Mark?
I'm wondering if that coral behind the Paragorgia is a type of bamboo
Sure.
coral.
I'm
thinking bamboo. It just
Yeah.
feels bamboo-like.
We'll come down to this ledge
But
and
I
look at
could be
stuff.
wrong.
I think your feeling is pretty accurate, so.
No, it's gonna be
Another thing that's
15 meters
interesting
at
to note right
1.5
now while we
degrees.
have this in the screen is to look at these
Yeah, it'll get us a
rock
little
walls
closer to
that
the
have
edge.
overhangs. And this is the kind of
Yeah.
oversteepening that conditions these kind of
Okay,
walls
Dan.
for slope
Sure.
failure.
A little bit.
When you're getting gradients in excess of 90 degrees, obviously the gravitational weight of the sediment is much larger than the shallower, gentler slopes that we were looking at earlier.
This overhang is really cool.
Yeah, it's again, I'm trying to get a kind of idea of the sediment
All kinds of
type.
stuff here.
It looks like a layer of
Wow.
much coarser material below fine grained material below. I can't
Thanks,
quite
Nav.
confirm that, but yeah, I'm trying to get an idea of the depositional context of this area going up.
25
And
meters out, so it's like
I think in this area, the only way to be able to do that, given all the incrustation biology on that, would be to take something like a core sample to look at the undisturbed sediment
look at all that stuff.
below
Most
and
underside
beneath
depicted.
within this rock wall.
Yeah, I just want to point out that on the left going up the screen, there's a bunch of those tubular sponges sticking out. So I wonder if that's what we were seeing before. They sure look like sponges here.
Yeah, Scott. We saw them briefly on the dive yesterday. These same, like, really long tubular
That fish
sponges.
has a
I think they're a type of
squid in it.
demi
Oh, yeah.
sponge.
Pretty
And
good.
in the previous dive, there were also very similar images of that tubular sponge.
Our co-pilot just noticed there's a little squid in that fish's mouth.
Oh, this fish caught a squid?
Yeah.
Oh, let's check it out.
That's why it looked so funny.
Watch my upper swing arms
So
there.
it looks
Yeah, I
like one
think
of
they're
these
clear.
cutthroat
Okay, Art,
eels, the
why don't you do a partial zoom?
Synaphobranchus gracilis, has caught some
Go a
sort
little
of
tighter.
food item, perhaps a squid.
Go tighter if you got it. Hold that.
Oh, oh, oh.
Lunchtime.
Yeah, we didn't see the predation event
Cool.
itself,
Okay,
but we
come
certainly
out.
saw the evidence of it. That guy is heading home with a full belly.
Well,
Okay.
that was a pretty cool predation event. But poor little squid.
Okay.
Oh, there's another one of those geodia sponges that we just collected
Okay.
on
Sounds
the
good.
wall, along with a number of these flame scallops. And there's a lot of single
It's like
polyp
I'm on the move a little bit here.
sclerotinians
Okay.
all over this
Nicely
wall,
into position.
along with small
Are
demasponges
you
and
with us till the
crusting
end, or are you gonna go out
sponges,
on deck?
plate-like
I think
sponges,
I'm with you.
leaf-like sponges. It's a very spongy
Megan, as we
ground.
were coming down here, we passed something that was a pretty bright yellow. It's off to the left. Have you looked at any colonies today that were overgrown with yellow zoanthid?
No, we haven't seen a colony overgrown by zoanthids, but I did spot that one, and then we got distracted by the eel.
Right. Yeah. Okay. So there's a couple of things to the left.
And I think we're seeing some Lophelia, this white sort of zigzag coral.
I did not. Okay, thanks.
And Pilot, the coral that we were talking about is this bright yellow one. Could we get a snap zoom on that?
Sure. Just slowly saddling my way over, and we'll zoom once I get there.
Thanks.
I still haven't seen the bamboo you were talking about. Were you referring to this zigzag one?
Oh, no, the one I was talking about was the really large fan colony that was right behind that Paragorgia we sampled.
I thought that was another Paragorgia. I thought that was a pink Paragorgia, but
No, it was too
Paragorgia.
fuzzy to be Paragorgia,
Be advised.
Copy.
but we are going to check it out.
Little vortex there.
Yep.
All right,
More
moment of
vortexes.
truth. Is this a parazoanthid?
Okay, we can go in, Art. Hold that.
I'm putting my money on that.
Oh, I think you'd be right. But then you get a photobomb squid.
After the requisite squid photobomb.
So, what we're looking at right now, this yellow, is
Sorry,
what
Art,
we
what was
call
that?
a parazoanthid.
Okay.
It's a type of coral that grows on top of other coral skeletons. And
Going a little tighter.
there's a lot of different ones, but this one is a bright yellow, and you can see how it looks different compared to the other corals that we've zoomed in on, in that there are a lot more tentacles on this coral than on, say, the Paragorgia coral that we just sampled that only has 8 tentacles per polyp.
We should bump it, and then they'll all go.
This one has a picnogonid sea spider on it, on the left side there. And I also, when I look at this, one of the things that I'm fascinated by is that this zoanthid has overgrown a skeleton, as you noted. It's basically a parasite, but it hasn't extended to anything else. It's only restricted to this one skeleton. I wonder why that is.
Going a little tighter if you got it.
Yeah, I've noticed that too.
Go ahead.
Couple squid inking back here.
Okay.
Great, thanks.
Sorry, Art.
Yeah, so my thinking here, Scott,
Starboard
is
vert.
that
Oh,
there
that's what's
are
going
a
on.
couple. The parazoanthids
Yeah.
are
Yep,
unique
there it
in
is.
that they just
to,
grow exclusively
Clear it.
on
Yeah.
Invert it.
the coral
do the
skeletons,
inversion.
but they
Oh,
don't
yeah.
grow on the rocks. And then other zoanthids will grow on rocks and may grow on coral skeletons.
Yep, and some will grow on sponges, and some will grow on crabs.
Actually, we've got
and so
the
on.
crab
So
eating a squid
it's
here.
really interesting.
You can go in,
It'll be,
Art.
kind
Yeah,
of a cool
fighting
study
over
to
Oh.
figure out what it is that's attracting the larvae of the zoanthids.
Crab's got a good hold on it, though.
Little fight going on here.
Yeah, the crab's like, no, you can't have my squid. And
I thought the cutthroat was going after the shrimp that was right by it, but maybe he was. But then he decided that squid mantle was a little bigger of a chunk. But if anybody has tried to get a stick out of a fiddler crab's hand on the beach or something like that, you'll know. But this
It's come out a little
cutthroat
bit.
seems to be pretty persistent as well, and we've got a good tug of war going on here for this piece of squid.
these cutthroat eels are pretty hardcore when it comes to feeding.
Would you say they're pretty cutthroat, Megan?
I would.
I want to say that, Jeff.
Oh, this is a free-for-all here. I'm wondering whether the eels are uniting on one team. The crab does seem to be holding its own. Not bad for 2-on-1. Yeah, this is a great tug-of-war that we're observing right here.
Okay, Art, you can go in a little bit.
Cutthroat eels are pretty ambitious predators in the deep sea, but go crab for keeping hold of your meal.
Now that the crab has fended off the other would-be scavengers, I believe he's luxuriously basking over his delicious calamari.
Oh, they haven't
Never
given up
mind,
yet.
eels aren't done. Boy, they are nothing if not persistent.
I'm pretty sure these eels are going to keep coming back around until they get their fill. No one passes up a good meal in the deep sea. Everybody loves squids.
The crab should have plenty to share.
Yep, but that's
Certainly
what
can't
one
eat
of our
all of that.
water column and squid experts, Mike Vecchione, says in the chat room that everybody loves squids, and I think we're fans of them as creatures, and also they're just universally thought of to be delicious.
On a sad note, we have only about 15 minutes left in our dive. But you can't
It's come
say
out a little
no
bit,
to
Art.
a good crab, squid, eel,
Hold that.
interaction.
I just brought hydraulics down. I still have them up.
Thanks. Let's see if we can zoom in on its mouth. When you're ready, Art.
Yeah, and this is another great example that we're seeing, like that lobster that was actually also eating squid tentacles instead of a mantle last time of those, I believe they're called maxilli, the little tiny, very fine motor eating tentacles, or not tentacles, kind of appendages near the crab's mouth.
They're called maxillipeds, so mouth arms, or mouth legs, if you will. And so crabs and lobsters have 3 sets of maxillipeds. The one you're most familiar with is the largest outside one, which is the 3rd maxilliped, and then they have 2 more sets closer into the mouth. Those are those small little ones that will chitter at you when you are talking to your crabs on the beach.
It's a great
As
shot.
one does.
I mean, you don't?
I absolutely do. And boy, you can really see
That's
how
what happens
effective
when
those
Art
claws
Howard's on the
are
stick.
in terms of just ripping off
Right.
chunks of that squid and then chowing
Turn
down.
a little bit to the right.
That's why these crabs need those maxillipeds along with their chelipeds, those pincing arms.
Yeah, and Mike Vecchione does note also in the chat room that it looks like the crab is kind of rifling around inside of the mantle and pulling kind of the innards of the squid out and eating that rather than the mantle itself. And that makes sense. The mantle is mostly muscle. It's probably pretty tough. I don't think you can get a nice braise going down here in the deep sea to kind of break down those tough muscles. So in lieu of squid asabuco, I think it's going for squid sashimi of the more tender inner variety. So good job, crab. You're a gourmand as well as a champion, fending off those would-be scavengers.
Nicely put, Jeff. It's
You might notice that the mouth parts have two parts.
Tell us more, Mary.
Well, there's an inner part that's used for feeding, which you can see quite nicely, but you'll also see a lot of fluttering, and those are the outer parts that are used to create the external respiratory current.
Oh, interesting. Okay, yeah. So one is for feeding, and then one is more for breathing. Gotcha. I see. Specialized. Thanks for that. Yeah, and that is really reaching deep into that mantle to chomp down.
It's probably a vortex.
You'll also notice those short twitching antennae.
Art.
Those are a sense of
A
smell.
little thruster issue to work on.
They were definitely twitching. It must smell awfully good.
Yeah, starboard. Hard to say quarter starboard, but yeah.
Yeah.
One of the uppers.
All right. You want more of that, or are we good?
I really want to see
Okay,
that
great.
big coral right behind the paragroid, Jeff.
Okay, we'll go take a look at that now. Sorry we got distracted there.
No worries.
Yeah, it was a
really
great
cool.
distraction. No regrets at all. That was fun.
It was exciting, informative. What more can you ask for?
Fine. Sounds good.
But I wouldn't feel right if we left without seeing this coral, because I'm just dying to know if I'm right.
So it's the reddish, brownish colored coral here?
Yes, it's this reddish brownish one.
I feel like
Okay.
you were right. Got that bamboo-y feel.
It sure does have that bamboo-y feel. And it's quite a large fan.
It's very large, and I find it very interesting that it's growing
Hey, Brian.
horizontally
Yeah.
from the wall and hasn't,
Welcome back.
sort of reoriented upwards. And I wonder if that has something to do with that big paragorgia
Upper
above it.
swing arm. Should be
It's got to
fine, right?
kind of move up to catch the currents that are sweeping around this rock
I
instead
think
of growing,
your brows perhaps.
basically, against gravity.
Yeah, that would make sense.
It's also, Jeff, I wonder, this must be a pretty stable substrate, because these are pretty big fans that are kind of perched on carelessly on the edge here.
Yeah, that's a good observation, and it's something that I speculated
Brian,
on a
what's
fair
going
amount
here?
yesterday. We kind of got from our Canadian colleagues, and I'm assuming the geology here is broadly similar, an idea of the minimum age range of the sediments
Go ahead
and the
and do a
rocks
tight-
that this canyon is
tight
carved into, and that would have been in the approximate age of Pliocene, so about 4 million years or older. So this is really consolidated stuff. But it also has a lot of mechanical strength and
Show
a
the headlights.
high angle of
Ready
repose,
to turn up some light for you.
unlike some of the other wall sediments that we saw
Lights coming out,
below
3,
and
2,
above this formation, which were more gently sloped backwards, which meant they were more prone to failure and gravitational oversteepening
copy.
than the sediments that we're on right now that form these sheer walls.
So this to me looks a lot like, I think, the specimen you collected on dive 2, I think. Dive 1 or dive 2, you collected what you called a nodal branching keratoisidinae. I think it looked like this.
Yeah, that was the last collection from Dive 1 at the Gully Canyon.
Okay.
And you might find this interesting, Scott. When
Sure.
we preserved it in ethanol, it turned
We'll stick with
black.
this. They're really interested in this
Yeah,
coral.
so I wonder if you would even before preserving it, it might have turned black. Did it also kind of slough off the skeleton?
No, the tissue stayed on the skeleton just fine. It was just really, it just turned to a really, really dark black, and we changed out the ethanol, I think, 2 or 3 times. It was just that dark a color.
Yeah, so that's really interesting, and we see that in some of these bamboo corals, and it's got to be related to something going on with secondary metabolites, and it'll be interesting to see if that's got some kind of genetic relationship. Only certain bamboo corals do it, and there's a genetic basis for it, or if it's something independent. Boy, these are chock full of, you can see in some of these polyps in the center that are pointing downwards, there's a lot of these rod-like sclerites. And there's a few needle ones that are sticking between the tentacles, but not very prominently. I'm a bit flummoxed right now whether to tell you this is something like Jasonisis or something in the B clade. It's going to be one of those two. But a really interesting specimen, so I'm glad you got a collection of it the other day.
Yeah, go ahead and zoom out.
So for the rock jock, just to confirm, this is a bamboo coral?
Yes.
Yeah, I think this is a
Okay.
Well, I haven't seen any
Thanks,
nodes,
guys.
but I don't know what else it could be.
See any nodes, Dan?
What's up?
You see any nodes in it?
If you get a
Branches,
chance,
branch
can you
points.
get a picture from behind of that squat lobster-looking thing? That could be something pretty rare.
Squat lobster.
On top of the colony?
Yeah, that's it.
Top of the colony. Oh,
Oh, okay,
That-
I see the arm sticking up. Pilot, do you see that little squat lobster on the
Yeah.
top?
What if you just pivot
that's fine.
starboard?
Yep. I can hold this for now.
So my first thought with those long arms is that it's a
Do a partial zoom?
chiron-stylid squat lobster. Is that right, Mary?
Hold that.
It used to be. It's been put in a different family called the Sternostylidae.
Okay.
Copy that.
Oh, that's interesting. New taxonomy for me to learn.
Yeah.
But you'll notice it has those enormously long legs, and the front legs tend to be bent at some weird angle, which this one certainly has.
I think I just got a little
Oh,
vortex.
there's 2 of them.
Starboard.
Yeah. Sorry, Brian, we got a little thruster
Yeah, I
issue.
didn't think its legs were quite that long, so there we go.
I think we just got a little sample in the starboard box. I gotta clear my thruster here. Okay, let's come back in.
What I was doing, Dan, is like every time I would impulse, I would look and see if both bars achieve the same percentage. You know
On the
what I mean?
feedback.
So what do you
Yeah.
think,
So if
Mary?
one's low, one's at 100, then you're like, oh,
Well, I'll have to have a look at them later. They can be identified by the spine arrangement on the back.
starboard
So
axial.
it's pretty spiny along the arms and on the back, and the spines seem to be colored more of an orange color compared to the rest of the body, which is
Just
more
hold
white.
it there.
Yeah, and just a
I just need a
body
few frames of
to
it for detail.
light ratio
Rather
that
than
almost looks more similar to a
little
sea
thruster
spider than
issues
it
here.
does some of those more squat and stout squat lobsters. Very interesting.
Well, I'm hoping we got a few good frames for you here. Let's come out. Brian.
Unfortunately, we know very little about feeding or behavior of these animals. Most of them are known from just dead stuff.
Sure.
Oh, okay. Yeah, every time I've seen spot lobsters like this, they're perched in these large fans and with their arms up
I think we're gonna have to start
and
coming
some
up here
sort
pretty
of
soon.
like feeding posture, perhaps.
Okay, Brian, let's try again.
Could be. But these animals tend to hang out in the larger sea fans rather than in the little wiry things down on the bottom. But these animals tend to hang out in the larger sea fans rather than in the little wiry things down on the bottom.
Okay, Copilot will go ahead and get everything stood, and we'll come up when he's done with that. Let's go ahead and zoom in, Brian.
So we're going to try to get a few last images of some of these squat lobsters before we leave bottom. We are at the end of our dive.
Sure.
We always seem to end on these really exciting things. It's such a bummer.
Okay.
I also think we're spotting some barnacles with black feeding
framing.
appendages
Okay.
in the background.
Focus, but
I just
Okay, let's
randomly
come out.
remembered that those are
You
called
got a few
cirri.
frames for her.
Yes.
How did,
I'll turn
where
my fog lights
did,
off.
what
Okay.
dark
Yeah, let's go
corner
ahead
of
and
my
start
mind
getting
was
set
that
up. You
hiding
wanna
in?
call it now?
Jeez.
So I saw those too, but they looked like they were coming out of the tubes.
Just let Watch Lead know we're, yeah.
So are we sure they're cirri and not Seriantid anemones?
Okay.
Well, when I saw them, it looked like they were coming out
Go ahead,
of
Nev.
plates.
Yeah, I agree, Megan. It looked like they were, moving like a barnacle cirri, but
Could you repeat that?
sure.
Sorry.
I thought they were coming out of those tubes that we were seeing.
Okay, great. Yep, no problem. Appreciate it.
Go ahead.
All right,
There
so
are some deep-sea stalk barnacles that tolerate mud quite nicely.
Okay.
unfortunately we are
So let's
preparing
we'll
to
just-
depart off
we'll
bottom.
get in
This
tow,
is the end
we'll
of our
discuss
sixth
that.
dive in Kinlan Canyon
Okay.
on the
We came up a little bit. Would you like
EX1905
to come
Like
up
2
further?
Deep
Yeah.
Connections
Healthy
cruise.
delta.
This was an extremely exciting dive. We saw all kinds of wild stuff. It was another Spongapalooza, all kinds of corals, some interesting geology. Really, what more could you ask for?
Well, we
because
got
to stretch
some
out, I'm pretty much going to be
you know,
going
Yeah, into
into the slope.
our squid photobombers. We got some squid eating action.
Saddle.
It was a
get
wonderful
more under
time
you.
for all
Gotcha.
kinds.
Absolutely. So as we're ascending, we are going to have our dive planning call for tomorrow's dive, which has been changed to Oceanographer Canyon, and that will be in about 10 minutes. So if you want planning for this dive, please stay on the line or get back on in about 10 minutes, and we'll go
Pretty
over the
much
details
lined up
for
the
that.
ship, so you're just going to come starboard, right?
So we're up in the water column now.
Ready for
So
me to do
this
that?
is your geology lead for the
Got a 20
expedition,
meter
Jeff
delta,
Mobelt, signing
going to
off.
auto depth.
And this is your biology
keep my
lead,
heading
Megan
in.
Putz